In my talk, I will be explaining a slightly different concept compared to the other previous talks. So what we are trying to achieve here is to get selective targeting to the brain after ischemic stroke by taking advantages of the blood-brain barrier disruption. So as I mentioned in my introduction, blood-brain barrier, as you know, is a major obstacle for the translation of therapeutics for central nervous system disorder because of this protective nature of the blood-brain barrier. However, now there is strong evidence from several neurological conditions that this barrier function is actually altered in several neurological disorders. And I have summarized this in more details in a review article that I published recently in Expert Opinion on Drug Delivery. But my talk here will be concentrated on the ischemic stroke, as there is now a very strong evidence that the nature of the blood-brain barrier is severely altered after ischemic stroke, resulted in hyperpermeability, which is modulated by both increase in the rate of transcytosis and also disassembly to the tight junction, which is severely altered as a result of the ischemic damage to the brain. So the question is then how to take advantage of this hyperpermeability that's induced by the ischemic stroke to enhance drug delivery to the brain by learning from previous successful stories from nanomedicine and other pathological conditions. And as you all familiar that one of the reason behind the successful historical translation of nanomedicine in cancer is the selective accumulation of nanomedicine into the tumor as a result of the hyperpermeability of the blood vessels in cancer. So as it has been explained previously, when nanomaterials travel in the blood, they selectively accumulate in the tumor either through fenestrated endothelium or due to open junctions between the endothelial cells, but also could be happens through vesicular transport system. So these mechanisms are exactly the same that has been explained to contribute to vascular hyperpermeability in the brain as a result of the stroke. So what we'd like to offer here is a new tools for enhancing drug delivery to the brain after stroke, taking advantages of blood-brain barrier disruption using nanomaterials. And specifically for this study, I'm using liposomes because of their rich clinical history and also because of the flexibility in their design and also the choice of the drug. So this brings me to the aim of this study. So what we are trying to answer here is, can we selectively target the ischemic site of the brain using liposomes, taking advantages of blood-brain barrier hyperpermeability? And if this is the case, what would be the mechanism of the selective translocations of the liposomes into the brain? And what is the cellular localization of the liposomes in the brain? So to answer these questions, we use either fluorescently labeled liposomes or radio-labeled liposomes based on the questions that we are trying to answer. And then we injected these liposomes into a rodent model of ischemic stroke, which is the middle cerebrally artery occlusion model. We injected the liposomes at different time points after the ischemia, half an hour, 4 hours, 24 hours, and 48 hours. And these time points were carefully selected in order to cover the two stages of blood-brain barrier hyperpermeability induced by the stroke. Where in the first stage you have, in the first stage you have increase in the transcellular transport through the endothelial cells. Then this will reduce over time, and then in the late stage you have both increase in the rate of the transcellular transport, but also disassembly to the tight junction proteins. So first we looked at the accumulation of the liposomes just 2 hours after the injections with IVIS imaging. And as you can see from the IVIS imaging, we have very selective accumulation of the liposomes into the ischemic left side of the brain at all the time points. On the other hand, when we injected healthy mice with the same liposomes, you get very minimum accumulation into the brain. Similarly, you have very minimum accumulation from the contralateral healthy side of the brain. So quantifying this fluorescent signal in the brain shows that the accumulation was significant when injected in the half-an-hour group and the 48-hour group, which again linked very nicely with the biphasic blood-brain barrier damage induced by the stroke. So this collectively telling us that indeed the selective accumulation of the liposomes in the brain is linked with this biphasic blood-brain barrier hyperpermeability. We looked at these results also histologically. So the yellow signal shows you the accumulation of the liposomes in the left side of the brain, and this was very nicely correlated with the areas of enhanced blood-brain barrier permeability, as you can see it from the brown signal coming from the endogenous accumulation of IgG in the brain that is normally excluded from the brain by the intact blood-brain barrier, but it gets just the chance to infiltrate into the brain after damage. What was also more interesting to see is that in the early stage, after the stroke, the accumulation of the liposomes in the brain preceded any neurological damage. So the neurons here are still healthy, which is something very interesting for therapeutics, because then you can deliver a neuroprotective drug and preserve the functions of these neurons before they started to die. We have also looked at the selective accumulation in real time with the SPECT-CT images. We concentrated here on the half-an-hour group and the 48-hour group because these are the two time points where we get the selective significant accumulation. So during SPECT-CT images, the selective accumulation in the brain become more apparent 24 hours after the injection, since immediately after the injection, you have a high level of the liposomes in the blood that resulted in an equal signal from both sides of the brain. However, as the concentration of the liposomes in the blood started to drop, then you start to see the selective accumulation only in the ischemic side of the brain. Quantification of these images again indicated that we have significant increase in the ipsilateral ischemic side of the brain. On the other hand, we have no significant increase in the contralateral healthy side of the brain. And we also observed that there was no significant enhancement in the clearance of the liposomes into their cerebrospinal fluid. So this is also telling us that this could be one of the reasons why we maintain high signal of the liposomes in the brain even 24 hours after the injections, because the clearance was also very minimum. So now that we have proved that we could selectively target the ischemic side of the brain, taking advantages of blood-brain barrier disruption, our next question was to identify what's the mechanism of this selective translocation into the brain. So first we wanted to know if this selective translocation is actually linked with the enhanced and intracytotic transport through the endothelial cells. So to answer this, we labeled the endothelial cells of the brain, and also we labeled the CAV1 antibody, which is one of the components of these cavuli vesicles, and we measured their colocalization with the liposomes, which is explained here by the red signal. So you could see half-an-hour group shows that there is a high expression of CAV1 in the brain, and this was nicely colocalized with the areas of accumulation of the liposomes in the brain. On the other hand, you have very minimum expression of CAV1 from the healthy mice, and there is also minimum or no significant accumulation of the liposomes in the brain. And similarly, this was also observed in the 4-hour and 24-hour groups. However, CAV1 expression then started to pick up again at the 48-hour time point, and again the liposomal accumulation started to enhance in the brain in the late stage. So this is again confirmed this biphasic increase, and also telling us that this biphasic selective accumulation of the liposomes in the brain is indeed with this enhancement in the transcellular transport. We confirmed this result also with electron imaging of the brain tissues. That again is telling us that there is enhancement in the number of the transcytotic vesicles in the endothelial cells of the brain compared with the healthy tissues. And we also observed that these vesicles actually increase in size at the late stage after the stroke, and there was also morphological changes to the tight junction at that time point, which we didn't observe in the early stage after the stroke. And I will come to this point later. We looked also at the colocalization of the liposomes of the neurovascular unit 24 hours after the injection, and we could see that the liposomes still maintain significant colocalization with the neurovascular unit, with the endothelial cells at this stage, and this is something that we would like now to take advantage of in order to preserve the function of these neurons and also ameliorate the damage to the blood-brain barrier and minimize the infiltration of the inflammatory cells into the brain. Next, we wanted to investigate if the selective accumulation into the brain could also be mediated by the paracellular transport that's happening in the late stage after the stroke, especially that this opening to the tight junction, the size of the opening of the tight junction is usually larger than the size of the liposomes that we use in this study. So to answer this question, we implanted the cranial window into the mice and the barrel cortex, and then we used multiphoton imaging to follow the accumulation of fluorescently labeled liposomes into the brain. We have also injected 2 tracers, dextran, which is a marker for a paracellular transport, and also fluorescently labeled albumin, which is a marker for the transcellular transport. So normally, dextran is excluded from the brain parenchyma by the intact tight junction. However, damage to the tight junction that is happening 48 hours after the stroke resulted in a significant infiltration of the dextran into the brain parenchyma. And this was very nicely correlated also with the areas of the liposomal accumulation into the brain parenchyma, and you also see some uptake by the perivascular macrophages very early on after injection. On the other hand, albumin, which is transported by the transcellular route, enhanced in the both time points. So we confirmed this result also ex vivo with IVIS imaging. That's also showing us that dextran increased significantly only in the late stage after the stroke, which is mimicking the changes or the compromise to the paracellular transport, while albumin is happening in both cases, and in both cases we co-localized with the selective accumulation of the liposomes into the brain. So these data collectively suggested that the delayed accumulation of the liposomes into the brain could also be mediated by the compromised tight junction. So in addition to co-localization of the liposomes with the neurovascular unit, we also wanted to follow the distribution of the liposomes into the brain and see which cells are actually taking up these liposomes. So we have screened different types of cells, but I'm showing you here astrocytes and microglia. So we observed very minimum uptake of the liposomes by both astrocytes and microglia when injected just half an hour after the stroke. However, when we injected the liposomes 48 hours after the stroke, we see a significant increase by the microglial uptake. And this was observed both in the core of the ischemia and also in the ischemic penumbra as well. Quantifying of these uptake shows that we have about 40 to 50% of the microglia co-localized with the liposomes, and we've also observed that this uptake is also linked with the stage of the activation of these microglia. So the higher the activation stage, the higher the uptake of the liposomes into these microglia. And we are now taking advantage of this in order to shift the polarization of the microglia from M1 to M2 in order to reduce the inflammatory responses. So this brings me to the conclusion of my talk. So I've shown you here that we are proposing liposomal drug delivery in order to enhance translocation of drugs into the brain, taking advantages of blood-brain barrier disruption. We have shown here that liposomal transport through the blood-brain barrier damage is mediated by stepwise recruitment of both transcellular transport that's followed then by the paracellular transport. And our data so far suggested 2 time points for therapeutic interference with the stroke, and also identified 2 distinct therapeutic interventions that we are currently exploring in more details. So we can either target the neurovascular unit in the acute phase or the delayed phase after the stroke in order to preserve the brain function, but we can also target the microglia and macrophages in the late phase after the stroke in order to shift the polarization of these cells towards brain repair. So at the end, I would like to thank all the people who have been involved with this study, and we open the study for discussion. Thank you very much for listening. Any questions? Yes, please.
Working the fluorescence dye labeled exosome for their delivery, and when I just found the delayed uptake, I was wondering, especially 24 or 48 hours, wondering in case of the exosome, it's all taken up by the liver, and liver is metabolizing, actually disintegrating all those exosomes. So the fluorescence dye liberated from the labeled one is going to recirculate. So in your case, how did you, I'd like to ask you an advice. So liposome is not metabolized disintegrated in the liver?
Well, there is some fraction that will go to the liver and spleen, but I would like to emphasize here, maybe I didn't explain it clearly, that the late time point, the liposomes were injected 48 hours after the stroke, so I didn't wait 48 hours after the injection to see their accumulation into the brain. But I injected them 48 hours after the stroke, and then I looked at their accumulation into the brain just 2 hours after the injection.
Okay, thank you.
Any other questions?